All right, guys, we're going to get started off with our genetics unit with the introduction. If you want to open up the PDF file, which has the exact same slides as we have here, you can follow along, or if you just want to take a look at the video, that is all right as well. Just make sure you take your notes in your notebook if you're not able to print out the PDF, okay? So today we're going to take a look at DNA. We're going to take a look at the structure, and we're going to take a look at a little bit of replication, but we are going to focus on replication in our next lesson. And for our success criteria, we're going to be able to identify the structure, we're going to be able to pair nucleotides, and we're going to understand a little bit about genes and proteins and how they play a role in our DNA sequence. So to start off with, we know that we have genetic information and they come from our parents, and these are inherited characteristics that you get from your biological parents. Now, these characteristics, we call them traits. We're going to talk about traits later on, maybe in a week or two, when we look at Punnett squares and how these traits are being passed on. But as for today, we're going to take a look at what DNA is. Now, you should know from previous years that DNA means deoxyribonucleic acid, and these are molecules that contain genetic information. They get transferred between generations of organisms, and they can change the properties of a cell because they have the ability to code for what they make in your body. Now, these genes are the inherited information that turns into traits that can be shown on your body or on an organism's body. Now, for genetic material, they're formed into groups, and this DNA is what codes them into groups, and each little coding pattern will turn into that specific genetic material. So we break it down. In the nucleus, it contains a cell's DNA. Generally, DNA is in chromatin form. So chromatin form is a long, thin fiber of DNA and protein that's together. And we'll talk about DNA and protein together in a little bit, but these chromatin fibers, during cell division, they'll actually condense and they'll turn into what's called chromosomes. And you learned about chromosomes last year. When they go through mitosis, you're able to see them, but then as they go into telophase and cytokinesis, they start to disappear. That's because they go back into chromatin form. Your human body has 46 chromosomes, okay? And the earthworm has 36, corn has 20, mosquito has 6. So depending on the size and complexity of the organism, they will vary. Now, we talked about chromatin, and inside the chromatin is actually DNA that's tightly coiled around these proteins called histones. And histones are these things right here, okay? So they package with our DNA, and they get very wound up together, very tightly coiled, and that is to make sure that they don't get damaged. Now, if we take a look at a little bit of a history here, in the 50s, Rosalind Franklin, scientist, actually used X-ray crystallography, which is a type of X-ray that takes a look at molecular structure. And what happens is she found out that the DNA has the spiral or helix shape. And further on, other scientists like Watson and Crick further called it a double helix. Why is it called a double helix? Well, if we take a look at it, it actually has two separate strands that come together. And that double helix is because these two strands of their chemical composition is why they look like it's swirling around while it is in ladder form. So we call that double helix. Now, inside these double helix are long chains of subunits called nucleotides. They are created by two things. One is a sugar phosphate backbone, which is created by a sugar and a phosphate group. And they're ring-shaped sugar, also known as deoxyribose, and a phosphate group, as well as a nitrogenous base. And these nitrogenous bases will determine what the coding is in our DNA. Now, there are 4 types of nucleotides, and these nucleotides will come together to create that strand of DNA. Now, 2 types. One type is called pyrimidines, These are our single ring nitrogenous bases. The two types are within that are thymine and cytosine. Our purines are double ring nitrogenous bases, and they are adenine and guanine. Okay, we only have to know at this level that the structure creates one ring, and your purines will create two rings. Now, from those nucleotides, they will be joined by covalent bonds, and they get connected from the sugar to phosphate, to sugar and phosphate, sugar, phosphate, so on and so forth. That's why we call it a sugar-phosphate backbone. As you can see right here, it is repeatedly sugar and phosphate, but attached to each of those sugar-phosphate connections, we have our bases that are here. Now, it could be thymine, could be guanine, could be adenine, could be cytosine. And that brings us to what we call base pairs, because they're all complementary to each other. And that bond is what holds the helix together. And the spiral shape comes from the placement of these bonds. So exclusively, thymine will bond with adenine, and cytosine will bond with guanine. We call these complementary base pairs. So anytime there is a thymine, the complementary nucleotide that is going to be attached to it will be adenine. And anytime you see a cytosine, the complementary bond will be guanine, and it's the reverse as well. So if you see adenine, it's going to be thymine that goes with it. If you see guanine, it's going to be cytosine that goes with it. So these are our complementary base pairs. This creates possibilities of sequences that can go on forever. Now, these nucleotide chains can vary in length depending on what the DNA information it needs. So it could be longer, it could be shorter, but all in all, it makes up the code for the cell to create these proteins to have functions at the cellular level. So let's say a sequence of ATCGGA, it is definitely going to be a different set of information as CATGTTACC. Now we can see that there are different number of nucleotides here. Also, the order of the nucleotides are different. So that means informationally, they are going to be different. Now, the closer the two organisms are, the more similar the sequence is going to be. So how do we tell that in, let's say, DNA tests is for similarities between two people, whether or not they're related, how much of the similarities there are. If there's not a lot of similarities, then chances are they're not too related or not related at all. So we can also figure out the sequence of base pairs on the opposite strand when given one strand because of these base pairs. So when given one side of the DNA, because you know that our DNA is a double helix, so if we can see that our one side, if we know what the sequence is, then we should be able to figure out what is going to be on the other side because of our complementary base pairs. So if the sequence for DNA is ATCGAGTT, then our complementary base pairs would be TAGCTCAA. How do we know that? Well, because we just talked about it just now, about complementary base pairs. Let's go back. Adenine will go with thymine, guanine goes with cytosine. Okay, so that gives us information that if we need to put them together. So our genetic code, like the Morse code, is where DNA is stored, and it takes 3 nucleotides to create a code word, and each set of 3 bases is known as a codon. Now, because of the 3 bases in a set, and there are 4 different types of nucleotides, it can create 64 possible combinations. And therefore, DNA specifies how to put amino acids together in a particular order. This order is called your genetic code. Now, in order for your proteins to be manufactured, because your body actually uses protein, and your body requires a lot of protein. A lot of things like the creation of enzymes, creation of all these parts of your body, like your fingernails, your hair, and so on and so forth, is all proteins. Now, that has to be made, and it doesn't get made by the DNA. It actually takes the DNA and it sends a message using a messenger. It's called RNA. And RNA is very similar. They are codes as well, and they go into your ribosomes to create that protein. Now, in order for proteins to be manufactured, they have to be from the ribosomes. And DNA has to communicate with our ribosomes. So RNA now, its function also has 4 bases, but instead of thymine, it uses uracil. So if you can see codon here, if it has a U that is in his entire sequence, then you know that it's RNA. RNA is only single-stranded instead of double-stranded like the DNA. But what it does is it actually tells the ribosomes what to do based off the information it gets from DNA. And then it creates your protein chains that you're going to learn later on, either in grade 12 or first year, about how protein is structured. Okay, so we'll pause here. I know there's a lot of information, but I'm going to give you guys some time to digest it. So what did we look at today? We looked at the structure of DNA. We paired nucleotides to form a phosphate backbone, and we now understand genes and proteins and how they play a role in DNA sequencing.